So I'm excited to talk about something that I've gained a pretty big appreciation for as I've been working at Nikon and even previously. And as I've talked to customers and partners in the industry and academic areas, when it comes to applying imaging, there actually is an increasing need for access to imaging technology and for use of imaging modalities in research and development, but also screening and quality assurance. So it's become a more and more important part of the therapeutics development pipeline, but also after your therapeutics are developed, a part of the quality assurance process. So I'd like to talk about those two things today and give some examples of what this looks like. Of course, this is a very broad subject, and there's a lot that could be said for many hours, but nobody wants that, at least in this setting. So I'll stick with some examples that I think illustrate what can be done, and hopefully that'll get us started on a discussion and maybe some discovery for those who are joining us. So the first part of this discussion will be focused on microscopy in research and development. And really that usually encompasses phenotypic assays, and then some emerging model systems that are pretty exciting that I'd like to highlight. Then I'll talk a little bit about microscopic imaging post-discovery and how we can apply it towards product screening and quality assurance. So when we talk about doing imaging and screening in R&D, one of the biggest applications is something called phenotypic screening. And so when we're talking about phenotypic screening, of course, we're looking at a phenotype, and it includes connecting as observable traits, which is the phenotype, with a specific pathology or physiological response that helps us to identify disease-relevant parameters, right? And then we use those parameters to test for therapeutic agents. So small molecules, proteins, peptides, nucleic acids, other biologicals, even cells now as a therapeutic that can produce a certain effect, right? And so that's the screen, is we look for this effect on the phenotype. And this has been going on for a long time. One of the bedrocks of pharmacology is actually going back quite some time. This is a classical experiment done where cats were decerebrated, and then they were given different amine compounds, and the effect of those amines on the blood pressure was charted and recorded. And in fact, you can see here on the right-hand side is one of the charts. This was actually hand-drawn based on data that was being recorded. You know, nowadays we don't do it quite the same way, but it has origins that are quite deep in terms of looking at a phenotype and seeing how it's perturbed by adding chemical agents that are possible therapeutics. And this is how a lot of pharmacological agents were discovered and then eventually became drugs. In a more contemporary context, we don't use cats and dogs as much because we want something that is inexpensive, accessible, and ideally high content or high throughput. Those words I've highlighted in bold because they're very important when we're talking about screening, especially in terms of imaging. So high content means that the amount of data that can be extracted from each sample, each subject is high, therefore the high content, meaning that you can extract a lot of information per read or per round of experiments. And then high throughput means, if possible, you can get through a lot of samples very quickly, right? The more samples you can get through in a certain period of time, then the more efficient it can be. And so with that in mind, especially in the context of imaging, then the goal is to find an organism, a model organism that can be accessed easily and is not too expensive. And so typically these days that includes something like a rodent, so a rat or a mouse, and animals that are a little bit newer in terms of being used in this context, like a zebrafish. And I'll show examples of both. But in each of these, we can access the organs relatively easily. And in the case of the zebrafish, we can use many organisms in a small format, especially if we're looking at the embryos, for example. and they can be imaged easily. They're transparent. And so these models are gaining a lot of popularity. Some examples of what this looks like. On the left, we have a setup where a cranial window has been installed in a rodent, either a mouse or a rat. And this allows access to the brain for imaging. And using this setup, we can see what's happening on the cellular and subcellular level inside an animal as it's being stimulated or as drugs are administered. And this allows us to see in real time how the brain is responding to those agents. Another example on the right-hand side is the fish in a dish scenario. And in this case, it's small zebrafish embryos that are put into microwell plates. And these embryos can be imaged through the bottom of the plate or from above with a macro camera. But typically on the microscopy side, we'll look from the bottom with an inverted scope, and we can watch how the zebrafish embryos respond to environmental changes. This is often used now for developmental studies, toxicology, and other studies of that type, where this is actually even more high content because now we can look at hundreds of zebrafish embryos in these dishes using these wells as dishes are exchanged. And we can test a variety of environmental conditions. And we can see very well. So these are the biological models for doing screening in vivo. And what's nice is as the models are evolving, so is the equipment. And these are some examples from Nikon as far as what we offer in terms of equipment, but there are also other systems out there that do similar things. On the left, we have a multiphoton system. This is a point scanning confocal microscope that uses special multiphoton technology that allows imaging very deep into tissue. And so we can look and see what's happening far down into the brain as the animal is being tested. On the right-hand side, we have a high content system that allows incubation and imaging to be combined with robotics so that a schedule can be set up and many dozens of plates can be imaged over the course of a short period of time. This increases the efficiency of the experiments and allows us to do a lot of imaging experiments in one plate and extract a lot of data. And this is actually where imaging shines, because every image you collect can be used to extract a whole host of different types of data. And it can be archived and pulled out later to be reanalyzed as technology in terms of image analysis is developed. And so you have kind of a library, a nice catalog. And so especially in cases where we're using these microplates, we can collect a lot of data, so it's high content, and we can do many in an automated fashion, and so it becomes high throughput. You can also combine imaging of the animal with a behavioral camera, so we can watch the behavior of the animal and combine that with, in terms of using this system here, so that we can combine visual observation of the animal's behavior, so eye movements, twitching, those kinds of things, with imaging of the cells and how the cells are behaving to create an even more high content platform for getting more information from your experiments. So here's an example of what those imaging look like for the deep brain imaging with the multiphoton. So on the left, we see you can go very deep. This is going almost 1.5 millimeters into the tissue. And you can see very nice structure with the neurons coming in and interacting with different strata inside the brain tissue. And at each of these levels, like I said, we could look at different characteristics of the cells in that tissue and extract all kinds of different data. Here we can see 5 different types of data in terms of images that can be collected. And then in each of these, we can do things like count the number of nodes that are coming out of the cell body, measure the thickness of the stratum. We can look at the intensity of the fluorescence. We can count the number of bodies that are in the tissue to look at density. And this is a static image, but we can also look at dynamic images. So we can look at live processes happening. So here we can look at, in the top right corner, we see an example from Zhang et al. In a Nature Methods paper that's very nice, where they use multiphoton microscopy. and you can see the movement of cells through the vasculature. And so this shows that we can not only collect very deep data, but we can collect data in real time and on very, very small time scales. So that adds a fourth dimension to the data. Again, increasing the content that you can get and making it even higher content. Here at the bottom, we can see neuron signaling going on. And so this shows that, you know, technology has evolved to the point where we can collect a very rich assortment of data from the samples that we prepare for doing in vivo imaging. And we can also look at other organs. So this is a look at pancreas, looking at the acini after they've been stimulated with an agonist. And we can actually see how those cells are responding in real time. That kind of movement is actually the breathing of the animal as it's being imaged. And so the animals can be kept healthy and alive at the same time they're being imaged. And so we can see on a cellular level how the animals are responding. Again, a great example of doing imaging in vivo. Here's an example of looking at angiogenesis in zebrafish. And so this is another example of using a confocal microscope looking at zebrafish development. And we can see as these vessels are developing, the movement from you know, the kind of the asymmetrical movement of these vessels, and we can watch the cells as they extend these projections and interrogate the growth factor landscape, and it's helping them pattern in the proper direction. This can be combined then with you know, perturbations in the environment, addition of drugs, toxicants, these kinds of things, and then we can watch how those affect the development process. So another way to collect very nice, dynamic, high content imaging information. And then what we can do is we can actually measure this process by adding some kind of image analysis software. So this is an example of using NIS Elements, which is from Nikon. And we can actually overlay the data, and we can measure as these vessels are developing, their growth rate, their size, all kinds of things. So you can see once we've got this video, then we can take it and analyze it to collect all kinds
Melina.
of
Your screen
quantitative
might have
data.
frozen. I think your screen might have frozen. Can you hear me?
I can hear you, Melina. Thank you for letting me know.
Okay.
Is the screen frozen now? What can you see?
We are on phenotypic screening, deep brain imaging.
You might want to unshare and then reshare, Jeff.
Okay, let's give this a try. Thank you for letting me know.
Yep.
Sure.
Okay, can you see the angiogenesis now?
No. You want to start it over to, maybe I could have Sam share your screen. Sam, can you share Jeff's slides? It might just take a minute. You want to try it again, Jeff?
Yeah, let me try one more time here.
What slide were you on?
So I had made it to, what slide is this? Slide number 11 or 10, I guess, if we haven't seen the past
Sam,
the deep brain imaging.
Yep, Sam has your slide, so he could serve as your backup.
Okay, great.
That's why we always do a backup.
The slide?
Yes, that's the one. Okay, so I'll keep going with this slide. So in addition to being able to look in the brain, we can look at other organs as well inside of a live animal, right? So here's an example of looking at the pancreas inside of of a live rodent, and we can look at the acini after they've been stimulated with an agonist. And we can see which cells are stimulated and how many. And we can see this live. And as you're watching, you can see there's a little bit of vibration going on. This is the breathing of the animal. And so imaging data can be collected at a high rate that allows us to compensate for what used to be confounding factors like breathing, but also see cellular responses here as they're being stimulated with an agonist. Next slide, please. Here's an example using the zebrafish of angiogenesis. And there should be a video. And we can see that we can follow the angiogenic process. So here we see the movement of the new blood vessels as they're, the cells within the blood vessels are extending these projections and interrogating the growth factor space. And they're able to find their way towards the proper position by growth factor gradients that are patterning. And then in this system, we can perturb the system and test for toxicants or drugs that affect this developmental process. You can also look at genetic variants that affect how the vessels are patterning by altering growth factors, for instance. And this is a very powerful model that one can use by looking at these fish in a dish. And we can look at multiple fish, multiple genetic backgrounds, multiple perturbances, toxicants, drugs, whatever it might be. And then to increase the content of the data, if we go to the next slide, we can see an example of how these data can be analyzed. So here now a mask has been created that goes over the developing vessels, and we can use these to measure their rate of growth, their size, their shape, and we can extract all of those quantitative data and analyze those. And this is also a video, so this can be done in real time. And we can watch the movement of these vessels as they grow. So this is an exciting development where we can actually combine the latest in animal models, and we can combine technology to extract a lot of really informative and essential information from living systems. So if we go to the next slide, then we can see another example, and there should be videos here as well, I think. Yeah. So here's an example of a lab that I worked with actually in the past, where we're looking at zebrafish again, but we're looking at the heart, and the heart has been tagged with green fluorescent protein. And we can see here the beating of the heart, and we can create vectors that allow us to measure the speed of that beating and compare different genetic variants and see variations in heart activity across these variants. And then we can again add drugs and see how they correct for these variants. In this case, this is a variant that is seen in patients that have atrial fibrillation. And it's a subtle fibrillation that can lead to problems, chronic disease. And if we can model this in something like a zebrafish, which is done here, and then use imaging to look at the heartbeat and then add drugs and look at how that heartbeat is affected, then we can find treatments and therapeutics that go along that can be used to correct the symptoms and even the problems caused by these abnormalities. If we go to the next slide, then we can talk about phenotypic screening in vitro. So if we come out of the organism now and move into parts of physiological systems, including cells and tissues, then we can do even more high throughput and sometimes more high content as well, because we don't have to take care of an entire organism. We're just looking at a piece. And that can include a whole organ, but most of the time we're talking about cells in culture, and we're looking at cells. And this can help us get an idea of what's going on the molecular and cellular level. And so when it's time to really ramp up your screening program and do image-based screening, then usually, because it saves money and time, and creating a more efficient scenario, then we'll use cells in a dish. So if we go to the next slide, then there's always a discussion between target-based screening and phenotypic screening. So there is a target-based approach where you kind of take a reductionist approach and you look at one component of cell signaling, for instance. Look at a receptor that's connected to a signaling molecule, maybe like a GPCR, for instance, right? And you look at that and you look at its biochemical properties. But the problem with this approach often is that though it's very refined, it often does not give us enough information to see how our drug is going to act in the context of biology, right? And so that's where phenotypic screening really shines, is it's a more holistic approach that can look at the mechanism of action of your drug or your therapeutic in the context of the system that it belongs to. So instead of just looking at a receptor with its signaling molecules, we look at, in the context of cells, how they behave when that receptor is being activated or blocked, right? And so this is something when we're talking about screening, we should address and talk about the difference between these target-based and kind of more holistic phenotypic screening. And that's what I'll be talking about more during this presentation, for the rest of the presentation. So if we move to the next slide. So this phenotypic screening actually goes back to some development that was done almost 2 decades ago. So at the turn of the century, they were looking at making cellular-based screening and kind of coined the term cellomics. So I think there should be an animation here that highlights, if you just move, that highlights towards the bottom. I think just click on the slide or advance, should pop it up. So you can see, there's a lot of stuff here in this abstract, and we don't want to read all of it, but if you look, here they created a cytotoxicity assay that enabled the generation of 10 to the 8th individual physiological cellular measurements in less than 1 day. So this is why cells are so popular now in terms of phenotypic screening, because it's high content and it's high throughput. So it was done in less than a day, and it generated 10 to the 8th physiological measurements. That amount of data is crazy. It's crazy to think about, but it's also very valuable, right? And what enables this is images, right? You're able to collect thousands of images, and each one of these images can give you hundreds of data points, right? And this can all be done very quickly. And so if we move to the next slide, then we can see some examples of systems that allow us to do this kind of imaging. So on the left is an example of one of our biopipeline series systems called the biopipeline plate. And the biopipeline plate is for looking at fixed samples, and it holds on the right in that hotel there dozens of dishes that can then be transferred automatically onto the microscope and then imaged. And we can look at fixed cells that have been stained with fluorescent markers, and we can then use image analysis software to look at the size, the number, the shape, the connections between the cells. There's a whole host of these parameters that we can look at. Then if we want to add another dimension and look at cells over time, then we can look at a live cell system like the biopipeline live. And here now we can look at cell behavior. And this is what allows us to get even more content from our imaging, because frame to frame, as the cells are changing, as they're growing, as they're dividing, as they're interacting with each other, then we can extract even more information. And so the content goes up. And these are really the two main types of phenotypic cell-based screens, is fixed or live. If we go to the next slide, here's an example of looking at fixed cells using a cell-based screen. So this is looking at kind of zoomed in, and then we zoom out, and you can see that on the high mag level, then we can do things like count the number of cells, the shape of the cells, look at how many in each quadrant there are. And then as you zoom out, you get kind of a heat map. And you can see on a more macro level what's happening across a plate and across conditions. Again, an example of the power of imaging where if you look up close, you can get a certain kind of data. And then as you zoom out, you can get even more types of data. Another way that imaging is a very powerful means to do this kind of screening, where each well represents a different condition, a drug that's being used, or a therapeutic candidate. And then we can quickly extract a lot of information about how those therapeutic candidates are affecting the cells on the inside of these wells. So if we go to the next slide, here's an example of a method that's called cell painting. And this is a multiplexing method where cells are fixed and then they're stained with a whole host of different dyes. And these dyes target individual components of each cell. As well as others and their partners. And this method is very important because it allows for multiple parameters to be investigated. And then using a software platform, you can, this is called, in this case, Cell Profiler. You can then look at each of these images and you can create a library of phenotypic traits that can be interrogated at different times depending on the question you're asking. So a cell can be treated, for instance, or a sample of cells, a series of plates can be treated with a therapeutic candidate. And maybe at the time the question is, how does this affect cell death, right? So then you look and you see how many cells are dead or alive. But then maybe you might ask, okay, well, how's it affecting mitochondria? Well, since the cell components have been labeled, you can go back and you can look at mitochondrial shape, size, intensity of the dye, whatever it is, and you can ask that question again without having to run an entirely new experiment. You just reanalyze the imaging data. And so again, this becomes very powerful because we can create libraries of information that can be interrogated at any time based on the question that's being asked. So if we go to the next slide, then here's an example of using live cell screening to get information. In this case, the context is neurodegeneration and trying to develop therapeutics for this. So this is the reference. This was published in eNeuro recently. And if we go to the next slide, then this is the basic experimental setup. So using a system that we have called the BioStation CT, our partners at the Lee Rubin Lab were asking the question, how are neurons affected over time when they are introduced to a toxicant that mimics neurodegeneration, right? Neurodegeneration is a component to a lot of different diseases, diseases like Alzheimer's and Parkinson's, for instance. And so they wanted to model neurodegeneration. And so human neurons were created from human stem cells, and these neurons were then put in these dishes, and their health was tracked over time in the presence and absence of a therapeutic that would protect them against neurodegeneration. And the question was, can we find an agent that can protect these cells from degeneration in the presence of an insult? And if we can, how does it work, right? And so you can see there's a time course going on, and then 0 hours, 60 hours, 120 hours, cells were imaged. The same cells, the same groups of cells were imaged. And then a software algorithm was used to track these cells and give them a score based on their health. And so healthy cells were scored with green, unhealthy cells were scored with yellow, and sick and dying cells were scored with red labels. And the reason this is important is because this helps us now identify cell health over time. And so we can understand the difference between how therapeutic agents are acting. For instance, if you find an agent that increases the number of live cells by the end of your experiment, the question is, how did it achieve that increase in live cells? Was it because it preserved the cells that were already healthy from being injured, or did it make already unhealthy cells better? Now, the difference is important because we're talking about the difference between neurorestorative or neuroprotective, right? If you are developing a narrow restorative agent, that would be something you could give to someone who's sick to make them feel better. If you're developing a neuroprotective agent, that'd be something you'd want to give to someone to prevent injury over time, right? And doing a fixed time point kind of static imaging won't give you that information, whereas live cell imaging can. So if we go to the next slide, we can see what the data look like. So here we can look at a population of cells over a long time period and see how they grow. And you can see that in this image, there are green and yellow and red cells. And each one, like I said, is classified by the software as having a certain level of health. And we can measure that health over time in the presence of different compounds. So this is an example of what those data look like and how the software can classify those. And then if we go to the next slide, we see a summary of the data, so we can see which ones are healthy, which ones are unhealthy, and which ones are dead, and where they're going over time. So what cells start out unhealthy and become healthy, what cells start out unhealthy and become more unhealthy, what cells become dead. And then if we go to the next slide, then we can see how we can use advanced software tools that are even more advanced to do some pretty amazing things. So in the previous example, we saw how we can score cells over time and we can extract important data about their health in response to therapeutic agents. In this case, we can actually employ AI software, and our latest generation of software uses artificial intelligence that's very good at identifying patterns. In fact, artificial intelligence is much better than humans are because of the number of calculations that it can crunch through. And so if we go to the next slide, we can see an example of something we might call digital labeling, right? In this case, the cells haven't been labeled with fluorescence markers of any kind, no dyes, no fluorescent proteins, but we can use an AI algorithm that was created and trained on these data sets to find the nucleus and the cell borders. And just or maybe we don't want to. Maybe there's no label that we know that will tell us the information we need. So if we go to the next slide, then here's another example of how AI software can help us. Maybe there's data that was collected that was poor signal to noise. And now we want to go back and try to use those data. Or maybe the signal to noise is poor because of the sample. Maybe the sample wasn't able to be prepared with the best signal to noise, but we can actually use AI tools to remove the background. And if we advance the slide, then we can see that employing artificial intelligence, it can recognize where the background is and what the signal should be and remove that background and clarify the image a bit, clean it up. And now it reveals all of the cells, and we can now count those cells, measure their shape, their size, and make a lot of quantitative measurements that would be impossible if we still had that terrible background. And then if we advance to the next slide, then we have another example of what we can do with AI software tools. Here, we're looking at neurons. And if we advance again on the slide, we can see that networks can be found. And this would be very difficult for a human to trace all of these over spending a lot of time and effort tracing these and measuring their length. And it's also very difficult for a human to create a mask that would do this. But with the AI, we can train it to find neuronal networks, and then we can count the branches and the interactions, the nodes on the cells, and again, extract a lot of really important information from these systems. So AI is a really nice technology that's being employed now. And if we go to the next slide, then this is an example that I mentioned previously about of how advanced cellular models are helping us in our phenotypic screening that we do. In the past, cell models have been two-dimensional. It's been cells laid out in a monolayer, or sometimes using a transwell where there's cells on the top and cells on the bottom. And these are good, but they're not sophisticated enough to mimic human biology, for instance. And so more recently, models like organoids and organs on chips have been used in order to create a more relevant three-dimensional environment. And imaging is important in these models as well as the two-dimensional models. So if we advance to the next slide, here are some really nice examples of using organoids for screening and the different modalities that are used. Solutions that are available from Nikon as well as other vendors. But the exciting part here is that the technology is evolving to the point where we can collect some very nice high resolution data that can then be analyzed and quantified, right? And so, you know, three-dimensional systems now are coming into the realm of screening where they can be used in a high content and a high throughput way. If we go to the next slide, here's an example of an organ on chip. This is a kidney chip, for example, and you can see that in this system, one can recreate how a kidney is put together. So on the very bottom layer are endothelial cells that have been seated in a three-dimensional arrangement that mimics a vessel, right? And then there's a membrane in the middle, and then the next layer up, we have proximal tubule epithelial cells, and these are functional kidney cells. And here now we have an interface that mimics a functional region of the kidney where endothelial cells are interacting with proximal tubule cells, and you can flow in through that endothelial lumen there, different compounds, drugs, toxicants, and then you can look and see how this kidney model responds. If we go to the next slide, then we can see an example of what this looks like. So here we've imaged a kidney chip, and we start at the basal level, and we move through the lumen where it gets dark here, and then we hit the next level of endothelial cells, and then we move through and hit the proximal tubule cells. And this represents about 350 microns of tissue, and this is done using a confocal microscope. But you can see now we can look at this channel with a relatively high level of detail, and we can extract information across the lumen all the way up into the functional cells. If we advance the slide, then we can see that the resolution is very high. So we can look at structure within this and make measurements in terms of the size of these, you know, kind of artificial tubules that are being created. And then if we advance again, then we can even look at, and I think that should be an animation, the red one there. We can look at how the cells are arranged on the cellular level. We can count the number of cells as well as measuring the structure in 3 dimensions. So again, we get a very large amount of information from these systems when we use them in the context of imaging. If we move to the next slide, then we can shift gears a little bit. And we've talked a lot about how R&D is benefiting from imaging technology. But as therapeutics advance and we move into regenerative medicine, cell therapies, then the therapeutic itself is a cell, right, or a tissue. And so it's important that for these complex therapies, we have systems that are robust enough to interrogate and extract the information that are needed to decide whether or not we want to release a lot, or whether we need to go back to the drawing board, whether during a differentiation process we need to you know, halt early, or we can keep going, right? These are very critical. And even FDA has provided guidance saying that if we advance the slide, then morphology is, you know, especially quantitative morphology is something that FDA will accept when looking at approving these therapeutics and making sure that there's a robust quality control system set up. So if we move to the next slide, here is an example of doing that in the context of stem cells. So stem cells, especially creating human-induced and regenerative medicine, but it's not always a given that your iPS cells will be created, that you're going to create stem cells from your source material. And so it's important to be able to screen and identify actual colonies versus non-colonies. So an example is shown here. On the left is a true colony, and on the right is a non-colony. They're very similar. A human can tell the difference, but they do it very slowly and inefficiently. And so the goal is to create systems that can do this in an automated fashion. So if we advance to the next slide, we can see that this is indeed possible. So using advanced instrumentation and software, then the software can do at the same level of accuracy, and in fact improved accuracy and much better precision, what a human would do, and identify IPS versus non-IPS colonies. And this can be used on the R&D side, of course, for testing different derivation techniques. But once you've chosen your stem cell creation technique, you want to monitor that as you're creating batches of these cells and identify how efficient that is, and make sure that it's performing as expected. You'll notice here that these colonies can be identified by software. They haven't been labeled. And this is really important too, because if your cells are going to become a therapeutic, you cannot afford to adulterate them, for instance, by genetically altering them to glow. Because whatever your end product is, if it's going into a human or being used on humans, you don't want glowing human parts, right, being inserted. That's something FDA isn't really excited about. So in this case, we can use the software to identify these without a label. So if we go to the next slide, then we can see another example of this. Once the cells have been created, the stem cells have been created, then we need to look at their health. So if we advance again, we can actually see an example of what cells look like, colonies look like as they're seeding and growing. This is a challenging system because you can see there's a lot of debris floating around. But if we advance again, then we can see how software, modern software, is able to identify colonies, give each one a unique identifier, and then track their growth over time. And if we go to the next slide, then we can see an example of how this can be quantified, right? So on the right-hand side is a graph that's made. And if we advance one more time, we should see this moving over time. So we can see over time how these colonies are developing, and we can identify the growth rate. And even when they start to merge, we can identify different colonies and use that information to assess their health, their suitability for the next phase, which would be differentiation into some kind of a therapeutic. So if we move to the next slide, we can see that we can also identify their whether or not they're being differentiated. Again, without any kind of a label, this is all label free using advanced software. And the green is undifferentiated areas and the red is differentiated regions. And we can find colonies that are in this differentiated area versus undifferentiated state, and use that to make calls about the suitability of that batch for use in further downstream applications. Again, this is done in an automated fashion, and so it's unbiased, it's label free, it's in line. It's a very nice development, and it shows how imaging is very powerful in the monitoring the quality of what would be a cellular therapeutic. So if we go to the next slide, then we can see if we compare this with the gold standard of something like immunofluorescence, the correlation is very high. And so we can train software and automated systems to do something that's with very high fidelity and high accuracy when compared to something like immunofluorescence. But of course, like I said, it's suitable for doing it in line, so you're not destroying your sample to look at it. So if we move to the next slide, we can look at how this works in differentiation. So here we're making neurons and looking at those early neuronal progenitors and over time. So we're looking over the course of 8 days and trying to track how these cells are developing as they go from a pluripotent stem cell to a neuron. And an important transition state is a neuronal progenitor cell. And typically the cells are labeled with a fluorescent label, but again, that takes the cells out. They get killed basically as they're fixed. And so in this case, the idea is to identify those cells that are becoming neurons over time without a label. So if we move to the next slide, that's done creating, That's done by creating a series of masks that identify different progenitor cells. So if we look at the very bottom, then you can see along the bottom there's different colors that
Hey, Jeff, I think you froze.
Okay.
Can you-
Can you hear me now?
Now we can, yes.
Okay. So let's move to the next slide. And so we can see now in the context of a whole culture, on the top left image is just the raw data. In the middle is the data that's been analyzed with this digital staining, right? There's no actual label inside the cells. And then for comparison is the immunofluorescence. And the correlation is quite high. But what's interesting is that we can use this digital staining to look at over time the emergence of different populations. And in fact, amongst the populations that are stained in green, which is this nestin that identifies neurons, we can see there's distinct populations that we can't discriminate for between if we just use the normal nestin marker. They're all green, but among those green cells, we have different populations that we can pick up using morphology, for instance, density, shape, these kinds of things. And then we can follow their growth. And the graph shows that you have this purple, green, and pink populations. And as the purple population grows and becomes stable, then it gives rise to the green population, which grows and becomes stable, which gives rise to the pink. And these are all distinct populations. And this mimics development very nicely. So again, an example of how one can monitor the quality of a culture that's being differentiated from a stem cell to a neuron that might be used therapeutically by tracking the growth of different neuronal progenitors that are important in the production of those final neurons. And if we move to the next slide, then we can see an example of this looking at another process called epithelial to mesenchymal transition. And if you advance, then we can track this important transition as well. Then if we move on to something like more mature, like a mature neuron, then we can actually watch the, we can monitor the health of mature neurons by looking at important parameters, the soma, the area of the soma, the neurite length, the number of dead cells, and the number of interactions between cells. And we can look at this over time. Again, this is being done label free. So there's a lot of information in these images. And with the right tools, then that information can be extracted and used to determine the suitability of this culture, for instance, for use therapeutically. And if we move to the next slide, then we have a summary. So in general, we can observe biological responses to environmental stimuli and other alterations, whether it be genetic or otherwise. And this observation is really key to collecting data in the biopharma and biotech area. So phenotypic screens, whether in vivo or in vitro, allow observation to occur, whether it's at the organ level or the subcellular level. And we can look at this live in whole animals, or we can use cells to look at this. There are a lot of really exciting new models, three-dimensional models, organoids, organs on chips, that create kind of a human organ in a dish, so to speak, right? And these can be used with imaging to extract really interesting information. And then, modern hardware and software allows us to do 2D, 3D, 4D imaging and extract information even with unlabeled samples that is critical for assessing both the mechanism of a therapeutic as it's being developed, but then also the suitability of a therapy that might be in production, right? And these are applied to cell screening, manufacturing, these areas. And then I finally wanted to share a little bit about, if we move to the next slide, about some of the solutions that Nikon provides that enable users to do all of the things that I've shown in this presentation. So we have turnkey automated solutions. This is a new series from Nikon called the BioPipeline, and it combines our advanced imaging systems with automation. so that you can quickly and easily set up a system on a benchtop and have the kind of throughput that you need to collect high content data. So this is a really exciting product line, and it's something that Ruby can tell you more about, and also myself, and we're glad to show this off. And then there's one more thing, if we move to the next slide, that I wanted to share. So I think this is especially important in the context of what's being done at ULP, and also for a lot of people across in the California life sciences community. It's being able to access this technology. And I think what's at the heart of ULP, which is giving startups a way to access laboratory space so that they can do their science and develop their products. Nikon is happy to kind of join that same vision by offering a new product called the Nikon Bioimaging Lab. And this is a contract research product where we do imaging research for customers. So you can reach out to Nikon with samples, and we can do the imaging for you and send you back data that's been analyzed. Or maybe there's a question and you need us to help you develop an assay. We can do that as well. Maybe you have images that need to be analyzed. You can send us the images, we can do the analysis, and then send the data back. Our approach is to interact with customers on different levels and on different scales. So maybe you just have a small project, a few $100, maybe $1,000 project. Or maybe you have a larger project. We have customers in pharma that have long-term 6-month, $100,000 projects more, right? But we're set up to be able to provide the imaging equipment and software that is required to collect the kinds of data that I showed here, and to provide expertise. So it's one thing to have the instrumentation, it's another thing to hire someone that can use it. And we provide both expert biologists and microscopists to use the equipment and collect the right kind of data so that our customers can get the information they need to push their therapeutic development processes forward. And if we develop a nice assay, then we can deploy it on site as the companies grow and their screening projects get bigger. So this is something we're very excited about at Nikon. And that's really the end, I think, if we move to the next slide. So thank you very much. I appreciate it. There were a few technical difficulties which happened, but I appreciate everybody hanging in there. It's been exciting to share this with you. Please feel free to reach out. And I'm happy to take any questions if there are any.
Thank you so much, Jeff. Really informative and lots of information. I just wanted to go ahead and plug in at the really end to, it is part of our mission to assist startups in access of this, really cool equipment. And so we do have extensive partnerships with UCI as well. And I know Nikon has instrumentation at the UCI Stem Cell Research Center. I believe it's the inverted scope, the Nikon TI, and the dissecting microscope, the Nikon SMZ 1500. So if anybody would like more information about that, and please reach out. We're happy to make those connections for you on campus.